The need for highly effective, productive, and sustainable farming solutions has growers everywhere interested in biorational products. But as growers evaluate their choices, it's not only important to understand the difference between conventional and biorational products, but more importantly, the quality difference among biorationals. Let's take a look at the biorational products made from the bacteria Bacillus thuringiensis, or BT. Conventional insecticides act through a single mode of action because they depend on a single toxic component. Biorational insecticides, such as BT, are more complex. BTs have multiple toxin proteins and spores that all contribute to insect mortality. Taken a step further, conventional products can be measured in a straightforward manner by percent active ingredient. But for BT products, given their complex mode of action, percent active ingredient does not provide a good framework for comparisons. Instead, various levels of insecticidal components within the active ingredient are what determines product efficacy. The main components for BT Christaki strains are the Cry1 toxin series, Cry1AA, Cry1AB, Cry1AC, and the Cry2 toxins. When ingested, these toxins enter and bind to the midgut of pest insect larvae and cause cell death, rapidly inhibiting further insect feeding. BT products also include BT spores, which germinate inside the insect and contribute to mortality. Again, it is the level of the various insecticidal components within a BT product that determine its efficacy. Several factors combine to determine those levels, and most are related to the quality of BT manufacturing. There are three areas of variability that contribute to BT product quality. Let's take a look at each. There are thousands of strains of BT, but a relatively small number of strains are used commercially. These have been selected for toxicity on insect pests, which is driven primarily by cry-toxin protein quantity and profile. Every BT manufacturer uses different strains, and the level of insecticidal components produced in fermentation are unique to each strain. Quality in manufacturing, however, determines the level of BT insecticidal components in the end product, which in turn establishes field performance. BT is a living organism. The manufacturing process is complex. It involves growing the BT through industrial fermentation, and the ability of the manufacturer to control and optimize this process determines the quality of the product. Key factors include the ingredients. Growth media play a key role in BT manufacturing. Quality in, quality out. Oxygenation. The amount of dissolved oxygen available to the BT organism as they grow and multiply has a profound impact on toxin, protein, and spore development. Cooling. Fermentation is a heat-generating process, and cooling is required to maintain an optimal temperature for maximizing toxin protein development. Process monitoring and quality control. Just as knowledge and experience with BT fermentation greatly impacts each manufacturer's process, in-process monitoring and adjustment of all these parameters is critical to success. The highest quality BTs are also tested with insect bioassay throughout the manufacturing process to ensure the products are formulated within the expected product quality range. While an expensive aspect of manufacturing, this step is an indispensable part of delivering a consistent high-quality end product. To summarize, unlike conventional chemicals, BT quality cannot be predicted by just percent active ingredient, but rather by the BT strain-specific complex mixture of toxin proteins and spores of the formulated BT product. BT quality can be thought of as the sum of the following parts. 1. The BT strain being used. 2. The manufacturing process. 3. In-process testing, monitoring procedures, and traceability. Please join us for our next video, The Truth About BT Quality, Part 2, where we will look at real-world examples of BT products in the market. We will conduct chemical analysis of some of the BTs that are available and look at field data where that quality translates into yield and return on investment.